Welcome to Croplan's Alfalfa Virtual Tour. The purpose of this presentation is to introduce you to Forage Genetics' initiative to develop novel germplasm for the marketplace and to help educate growers and sellers on some of the basics of alfalfa physiology. Forage Genetics has a history within the industry for over 25 years. During this time, Forage Genetics has been a leader in germplasm development to bring high-yielding alfalfa varieties to the marketplace. FGI's research covers fall dormancies 2 through 11, and FGI tests their varieties across the nation as seen in the map of the US. Each dot represents a testing location FGI has used in the past decade. As we work to understand the effect unique environments have on individual varieties, we understand them better. As you can see, FGI's footprint covers the major alfalfa areas in the US. As a person considers the varieties available on the market, it is worth balancing costs with an understanding of the genetics. Many institutions, both private and public, can develop a variety of alfalfa for the marketplace. It is worth asking, does the developer believe in their genetics to spend the resources to produce the seed, or do they just sell the rights to the germplasm? FGI believes in the integrity of their products, and only the best performing germplasm is advanced for seed production under commercial release. Commercial varieties have been tested in FGI's product development platform to evaluate yield and quality potential. A second question you should ask yourself is, what kind of research is the developing company putting into their genetics? FGI takes an integrated approach to bring the very best technology to the market. FGI achieves this through native genetics and transgenics to help ensure their product's potential in the marketplace. Some specific callouts are FGI's Roundup Ready and HarvXtra technologies. More about these exciting technologies will be discussed during this presentation. Through this Venn diagram, you can see that FGI is a full service provider to the industry. They provide proprietary genetics, seed production, and best technologies to develop a product which is hoped will have a positive influence on your bottom line at the customer level. In the breeding process, it is important that we get things right. In general, it takes 9 years to get a new product from concept to market, and they require years of evaluation by trained professionals to identify these opportunities and new products. There are a variety of options in the plant breeding process, and generally, alfalfa breeding uses a process called positive selection. What this means is individual plants are planted in nurseries for approximately 3 years. During that time, trained breeders evaluate each individual on a myriad of factors, which include health, vigor, fall dormancy, persistence, structure, and quality, just to name a few. At the end of the 3-year period, the best looking individuals are selected and used as parents for the next generation of varieties, also known as SIN1s. SIN1s is a shortened term for synthetic generation 1. During this time, seed production plots are evaluated and negative-looking individuals are culled from the seed plots. The newly developed SIN1 seed is then used in forage yield trials, sometimes referred to as FYTs for short. During this time, the variety yield potential, persistence, and other potential production benefits are evaluated. At the end of the FYT cycle, the best performing varieties are advanced to SIN2 seed production and advanced testing. During advanced testing, the lead varieties are evaluated against industrially accepted varieties prior to release to the public as a new cultivar and into commercial seed production. Now that I have explained the general process for developing a new variety, I'd like to take a few minutes to describe some of the steps in more detail. While all traits are important, persistence is one of the most important because it directly affects the yield potential over the life of your alfalfa stand. Persistence is a catch-all term which incorporates a plant's ability to survive both biotic and abiotic stress in subsequent years. To ensure persistence, plants are grown in a variety of environmental conditions to improve abiotic factors such as cold tolerance. Additionally, plants are often selected from disease screens in nurseries to build pest resistance. Finally, Recovery after cutting is an important factor because it demonstrates a plant's ability to not only grow well, but to also develop a healthy taproot to ensure winter survival when the plants are dormant. Because of this, experimental alfalfa is cut on an aggressive schedule to identify individuals which have good recovery and the ability to survive long term. As mentioned previously, forage yield trials, or FYTs, are maintained for approximately 3 years. These block trials allow us to assess the persistence of the alfalfa in different environments while gathering key data for yield potential and quality in a variety of environmental conditions. It is important to note that the FYTs are managed under local recommended practices to simulate how the variety could potentially perform for the local growers. These practices include seeding rate, cutting frequency, and other recommendations such as fertility rates. When selecting an alfalfa variety, it is important to understand how many cuts per year are wanted and how many years an alfalfa stand is wanted to remain in place. Fall dormancy is a good metric for making this decision. Fall dormancy is defined as a reduction in plant height due to day length and temperature. Recovery, vigor, yield, and persistence are all related to fall dormancy as seen in the photo of a fall dormancy 3 plant variety compared to a fall dormancy 5 plant variety. As depicted in the graphic, generally, the lower the fall dormancy, the better the winter hardiness and the persistence. The trade-off is a lower annual yield, less vigor, delayed growth, and this results in lower forage yield potential overall. Alternatively, the higher the fall dormancy generally results in increased plant vigor, earlier maturity, faster recovery post-cut, and increased yield potential. The trade-off is higher fall dormancy varieties have lower levels of cold tolerance and are not ideal for cold winter environments. Thus, in the past, fall dormancy would give good predictions of winter hardiness. Today, with our nurseries, we're able to select based on fall dormancy, winter hardiness, recovery rate, production potential, and stand life, which more accurately represents fall dormancy and winter hardiness. This provides for better survival and higher yield potential. Continued progress in improving winter hardiness and persistence in higher fall dormant types has the potential to extend the growing season and increase forage yield. FGI also routinely selects for stem nematode resistance. These selections come from test evaluations, stem nematode screens, and from grower field selections where the pressure is present. The photo in this slide is courtesy of Dr. Kent Evans of Utah State University, and it demonstrates the impact of a variety with and without stem nematode resistance. The field on the right is susceptible, while the field on the left is resistant. Other diseases can cause problems, and they are part of the pest resistance package which FGI routinely selects for. But just like diseases in humans, diseases in plants are subject to evolution in their environment over time. Chemistries and/or genetics, which once provided protection, begin to break down and are ineffectual against novel pathogens. Two examples of this are resistance to Aphanomycetes and anthracnose. FGI is working with university researchers to characterize novel races of these diseases, which genetics until recently found in the marketplace have lacked resistance to. Through our selection process, we are developing robust varieties which can resist the historic and novel races of these diseases as demonstrated in our multi-race package. In the photo, you can see on the left a variety which is not resistant to the novel disease and an FGI variety on the right which is resistant. Again, in this photo, you can again see an FGI variety which is resistant on the left in this photo and a commercial variety which is susceptible on the right. Another benefit of FGI's genetics is the development of Roundup-ready alfalfa. Research has shown that such technology is correlated to higher yield potential and better feed quality while being convenient, allowing for flexible applications and increased savings to the grower. Since Roundup Ready's introduction in alfalfa at the turn of the millennium, multiple studies have demonstrated the utility of such a technology within a grower's system. In the early 2000s, researchers at the University of Wisconsin found that Roundup Ready alfalfa had a higher alfalfa yield and a lower weed yield when compared to conventional herbicide treatments and the untreated control. This has the potential to also directly improve yield because the bell will be composed of alfalfa as opposed to a mix of alfalfa and weeds. Reduced lignin alfalfa, commercially known as HarvXtra. Over the years, many alfalfa breeders and companies have worked to improve alfalfa's forage quality with limited success. One of the more successful attempts was performed by FGI with the development of the multifoliate leaf expression. A standard alfalfa leaf is composed of a petiole and 3 leaflets, as seen in the photo in the top right. Natural variation exists, which allows alfalfa to have 3, 5, or even 7 leaflets. While selecting for increased occurrence of this trait, FGI found a potential improvement in quality by 3 to 5% generally. While this was a small increase, it was an increase in the right direction, and FGI continued to search for improved quality traits. The quest for improved quality led to the development of methods to reduce lignin production through gene silencing. It was observed that reducing key genes in the lignin synthetic pathway could result in a potential increase of forage quality of 10 to 15%. It is important that we take a moment to first ask what is lignin and why lignin. Lignin is an indigestible phenolic compound in cell walls of plants, including alfalfa. It is responsible for cross-linking the cellulose fibers to add strength to the cell walls, but also reduces its ability to be digested. As cells mature, the lignin content increases, and this is why we see quality decrease with the age of a stand between cuts. It is estimated from ration models that a 10% increase in fiber digestibility has the potential to increase milk and beef production by 350 million per year and has the potential to decrease manure by 2.8 million tons per year. In an effort to achieve this dream of increased digestibility, FGI partnered with the Consortium for Alfalfa Improvement, which led Dr. Richard Dixon to identify the C-comp gene as a key target to reduce lignin production without affecting standability of the alfalfa in the field. Standability is also known as lodging tolerance. Just like rebar in a building helps hold concrete for wall production, lignin supports cell walls similarly, but the amount needed depends on the plant. Through experimental observations, FGI has found the right amount of lignin reduction while maintaining plant integrity and while improving quality. To date, FGI has not observed low lignin alfalfa to be any more susceptible to lodging than conventional alfalfa when compared side by side and controlled for environmental conditions. To help demonstrate this improvement in quality, imagine that we took 100 plants at random from a field of alfalfa and measured their quality. In this example, acid detergent lignin, or ADL, which is a component of fiber that is essentially indigestible. The lower the value, the more digestible the sample is estimated to be. Looking at the results of the 100 individuals, it is found that the distribution of the quality measurements are normally distributed, or rather, the variation occurs equally from the average of the total sample. As breeders, we look for these differences and try to select plants on either end of the spectrum depending on the target. In this case, we would want to select from those with low ADL. Over time and with extreme effort, modest improvements in quality are potentially possible. The benefit of FGI's HarvXtra trait is further demonstrated in the graphic. If 100 individual plants are selected and measured for quality in the same way from a HarvXtra variety, we find the data is still normally distributed, but just more tightly controlled. This means there is less variation in the sample generally. Further, we observe that the use of genetically engineered traits to reduce lignin has a great effect to improve quality more quickly than the use of conventional methods alone. As breeders, we are still able to then use conventional methods to select the outliers for improved quality to further push the quality improvement over time, as demonstrated in the shaded blue area on the graph. Alfalfa growers of today have to make a trade. As lignin increases with the age of the alfalfa between cuttings, the yield also increases. This leaves growers with having to make a trade-off: increase quality or increase yield. This decision depends on the specific operation and the goal of the grower. The question ultimately is, can I sacrifice quality for improved yield, or do I sacrifice yield for increased quality? So what makes HarvXtra alfalfa so great? The trait of HarvXtra allows alfalfa to be modified to reduce the lignin in alfalfa beyond what is possible with conventional alfalfa, giving growers the freedom to either one, maintain a normal harvest schedule for higher quality, or to delay their harvest 7 to 10 days for increased yield potential without sacrificing their forage quality. The first option is to maintain your cut schedule. Farmers can maintain a normal harvest schedule and achieve a higher quality. Let me explain. Forage yield trials, while tracking quality over time, have found that over the course of the life of the alfalfa between cuts, HarvXtra alfalfa has on average 10% higher quality as measured by NDFD. In the graphic, we see quality from 20 days post-cutting to 37 days post-cutting. Routine practice is to cut on a 28-day cycle to balance yield and quality. HarvXtra, when compared to conventional alfalfa, the black lines compared to the green, at the same 28-day interval, has the potential to provide approximately 10% more quality as seen in the yellow line on the graph. Option 2 is to delay harvest. A grower may choose to delay harvest for up to 7 days for higher yield without sacrificing quality they would have realized if they had harvested at their normal harvest interval. Again, with the same graphic as before, if we add in the yield potential curve with a blue line, we see that the yield is roughly maximized about 37 days after cutting. This is the potential of about 20% more yield depending on the variety and the other environmental factors. Following the yellow arrow as well, it is important to note that the quality at approximately 35 days is the same as the quality for conventional control alfalfa at 28 days. HarvXtra then has the potential to increase a grower's yield while preserving the alfalfa quality. Another benefit of utilizing HarvXtra in a farming operation where cuts are moved to 35 days as opposed to 28 days is the reduced stress on the individual alfalfa plants. The added growth time allows the alfalfa to store more resources in the taproot, thus increasing the overall health of the plant and the potential to survive the following winter. It has been observed that this results in increased yields over the life of the stand. During the first year in trials in West Salem, Wisconsin, it was found an approximate 15% increase in yield. This increasing continued during each year of the study for an average 30% yield increase over the course of 3 years when compared to the Roundup Ready alfalfa cut at the standard 28-day interval. It is important to note that this observation for yield improvement can be seen with any type of alfalfa, whether it is conventional, Roundup Ready, or HarvXtra, but in the absence of HarvXtra, the quality will not be preserved. A mixed approach for options 1 and 2 have been reported from early adopters of the HarvXtra technologies where they like the flexibility to cut their alfalfa while balancing weather, and operational and/or family issues with events. Another example where delayed harvest has had positive effects is on research forage yield trials in Touchy, Washington. In the photo, during the second production year, it was found that the treatments on the right had 35-day cut cycles. Those on the left were cut on a 28-day cut cycle. The yield on the first cut was not only higher for the 35-day treatment, but the recovery health of the plants was visually better as well, as demonstrated in the photo for the 5-cut versus the 6-cut harvest system. Again, the benefits of utilizing HarvXtra in this type of system allows for the potential to maximize your quality at the same time while maximizing your yield.